Right, so in structural biology we're trying to understand the structure of proteins, because the structure of proteins is inherently linked to the function. So the cell, inside the cell there's thousands and thousands of proteins, and if they function properly the cell is healthy, therefore the tissue is healthy, and therefore the organism is healthy. So if you imagine each protein being like an instrument in an orchestra, like a violin for example, the structure is really important because the way it's constructed affects how warm it's going to sound, you know, what noise it's going to make, right? Now also imagine if there's some small defects in that violin, right? Or if the neck is a bit bent, what's going to happen is it's going to start making some not so nice sounds. And that's basically what happens in protein misfolding. So when a protein misfolds, it will then start adopting a wrong conformation, and that can then means that a lot of them start sticking together, and then eventually, you know, you form these big aggregates, and then that causes the cell to die eventually and disease. Now, why this is important is because it underpins many very devastating diseases like Alzheimer's, Huntington's, type 2 diabetes. You know, collectively they affect 500 million people, you know, about 1 in 17 of the world's population. But the underlying mechanism is exactly the same. You have a normal protein that somehow misfolds and then it starts aggregating. Now this has been really difficult to study with traditional structural biology methods and other analytical techniques. So we've been trying to do this for about 15 years, specifically this protein called alpha-1 antitrypsin. So this is a protein that you've got in your body, and it's really abundant, but when it misfolds it gets trapped in your liver, but then it can't go to your lungs to do its normal function. So the patients get both liver disease but also lung disease, right? So it's very difficult, as I said, to study. We started about 10 years ago looking at it with the original SYNAPT. We were also really interested in looking at the real disease variant, the protein we could isolate from liver transplantation experiments, and so coming with a SYNAPT, a few years ago we had really early access. This enhanced sensitivity did allow us to basically look at the protein, look at these early misfolded intermediates that had not been characterized before, and also prove what the structure of the dimer is. And this is really key because this is the first event in the eventual polymerization pathway. So yeah, we're very happy. So, as I said, these proteins exist in sort of multiple conformations. So, even with the development of deep learning methods like AlphaFold, which predict the structure from a sequence, these states are not predicted, right? So you have to measure them. And because they're so very fast and interconverting, not many other techniques can actually look at them. So it's really important. Our North Star has always been to try to use these tools and the methods that we develop to answer real biological questions. So, over the last 10 years, we're focusing more and more at looking at the real, biological, not purified proteins, but so in our JAX paper, what we did is we isolated alpha-1 antitrypsin from that we isolated from a liver, because in alpha-1 antitrypsin, in very severe cases, the only cure is to do a liver transplantation. It's really important. If we really want to understand these diseases, which are very complicated, and you have got thousands of labs working on them, it's really critical that you have a whole ecosystem working on it. So you need the instrument manufacturers pushing the technology of sensitivity, resolution, dynamic range. You need people like us that like to drive the application to the biology, but then also you need the clinicians to collaborate to get as I said, the clinical expertise, the real samples, and then further, in the future you also will need people to translate that and develop therapeutics. So yeah, it takes a whole ecosystem to do this. So we still work on alpha-1 antitrypsin. So one of the other things that we are doing is we're not only interested in how the first bad notes from the orchestra come out, but we're also interested in is what happens to the rest of the orchestra when these first bad notes come out. So we want to see how the cell responds to these misfolded intermediates. So we've got a big program on that, but also we've got, we're applying the similar methodology to look at amylin, which is another peptide that is found in patients of type 2 diabetes. And also we've got a really big program on Huntington's. And there what we're looking at proteins that are implicated in expanding the trinucleotide repeat that's important in creating the toxic Huntington gene.